This is Francesca. She's grown to become a young, beautiful, and inspiring adult studying biomedical sciences. But she started to wonder how the inside of her body, her organs, had grown along with her. How do the intestines grow? The answer we don't know, and Francesca wants to find out. Luckily, current research has looked into the maturation of the intestines as a new field of interest. This is important to know because certain consequences correlate with your intestines when they do not mature correctly. Short-term effects are defects in newborns such as necrotic enterocolitis or infections. Long-term effects are malabsorption disease and inflammatory bowel disease. But maturation of the intestines can be regulated by certain factors such as glucocorticoids, for example dexamethasone. Glucocorticoids can accelerate maturation seen in fetal mouse organoid models. LGR5 is a gene which is expressed by these stem cells and can therefore initiate differentiation. So crypt villus organoids, small models of intestinal organs, could be made with these stem cells. This is an organoid being filmed under the microscope for about 8 hours. You can see the organoid moving due to new crypt formation. Studies about intestinal maturation have already been done in mice, but not yet in humans. Markers of intestinal maturation in vivo are lacking. So with this project we aim to, first, obtain gene expression profiles of human intestine tissues. Secondly, assess intrinsic components of human epithelium using organoids. And finally, study the impact of nutrition concepts and infectious agents on intestines. Francesca started to collect intestine samples from donors in abortion clinics. She wanted to isolate the LGR5 expressing stem cells from the crypts and culture them with several growth factors within DMEM and Wnt medium. Wnt is a signaling pathway inducing stem cell proliferation. To do this, she first cut the intestine open and scraped away villi to make the crypts more accessible. She then cut the pieces of intestinal crypts into smaller pieces and incubated these with EDTA for 30 minutes. EDTA is a substance which causes the crypts to become more flat and accessible instead of in between the villi. The EDTA is then taken out and the crypts are washed with PBS. After that, the supernatant is collected and centrifuged to collect the pellet. The pellets, containing crypts, are then put into Matrigel and incubated for 10 minutes. Organoid medium, which is made of DMEM, a medium with nutrients and supplements, etc., such as glucose, and Wnt conditioning medium is added. The crypts with stem cells are then cultured with certain growth factors to cause proliferation and formation of a complete organoid. But what does Francesca want to do with these organoids? She wants to see if these organoids can mature in the same manner as in vivo intestines by looking at the genetic profiles. Certain genes have already been discovered in intestine tissues from fetus. They looked at the changes in these gene expressions during development of the fetus. Francesca wants to compare the gene expression of the organoids with the intestinal tissue genes as a reference to see if the organoids developed in the same way. She will use microarray, qPCR, and immunostaining to do this comparison. Thanks to the development of this stable in vitro organoid model, it will be possible to define both the intrinsic and extrinsic aspects of the human intestinal maturation. However, Francesca is a very ambitious scientist. She also wants to do future research using these cells from the organoids. From the organoids, single cells can be made and plated as monolayers on transwells to be able to perform functionality assays and check if there is a way to accelerate maturation by adding different compounds and looking at the effects. Why is it so important for Francesca to produce these organoids and do different experiments with this model? It is important to develop a human in vitro system as there is a lack of human material. Also, understanding human biology in health and disease through a simplified and reproducible model is crucial for the understanding of intestinal maturation.